Hepatocellular carcinoma, also known as HCC, is the most common type of liver cancer, accounting for the highest incidence and mortality rates for primary tumors. Existing treatments of HCC, such as chemotherapy, surgery, radiation, and immunotherapy, demonstrate limited efficacy and convenience. The most widely used chemotherapeutic agent, Doxorubicin DOX, shows high efficacy. However, it causes side effects including autoimmune-induced hair loss, vomiting, diarrhea, and fatal heart damage due to accumulation in non-target tissue such as the skin. Thus, the development of a carrier system for DOX that would improve targeting and therefore efficiency, as well as minimize side effects, is required. A recent study published in the International Journal of Nanomedicine successfully develops a dual-ligand targeted core shell chitosan-based nanoparticle that allows DOX to actively target HCC cells. This nanocarrier employed electrostatic interactions between negatively charged carboxymethyl chitosan-g-polyacrylate and positively charged dual-ligand. The results indicate that the nanocarriers enhance cellular uptake of DOX to HCC cells